and of course in Latin, virus is poison. And so you talked about how the exosomes come out of the cell, they kind of soak up the toxins, which is what I've been saying. And then they've got a little lock on them and they head off down to some cells. They take all the toxins away from the cell before it breaches that cell wall. So do you want to talk a little bit about that? Because, wow, have I been slammed for that one. So
yeah, no,
I'm
you're
feeling some
absolutely right.
backup here.
So, and I think it's really, really telling actually about the meanings of the words. Excuse me. So virus does come from the Latin meaning poison, and it even was used to describe, I think, snake venom back then. So, and I think really what we're seeing with all these illnesses that they call viral illnesses is that they're really caused by toxins. And so I had this idea about exosomes, which was really interesting, and how does it play into this whole scenario? So exosomes, as you mentioned correctly, they're little vesicles or like sacs of fluid that look exactly like what they tell us viruses look like under the microscope. And it's something that all of the cells in our body make. In fact, all mammalian cells make this, and probably plant cells as well. So it's very common. And we put these things out at a base rate all the time. But when we're exposed to a toxin, or we have an acute illness, and it could be almost anything, like even ionizing radiation, it could be psychological stress, it could be pneumonia, asthma, it could be various poisons like pesticides or heavy metals or antibiotics, that our bodies put out more exosomes, and this is called like induction of exosomes. And the exosomes generally are thought most likely to have a communication function, that they basically communicate information that's contained in the genetic material to other cells in other parts of your body, and maybe even to other organisms outside of your body. But they have like specific receptors that target them to a certain type of cell, and then they arrive at that cell and give the information. But they've also been shown that they can actually eat up toxins outside the cells. So a cell would excrete them, and in the presence of toxins, like there was a great experiment done where there were bacterial endotoxins that would kill the cells basically. And when they mix cells with exosomes with the bacterial endotoxins, the exosomes gobble them up and the cells survived. When they mixed other cells that weren't expressing exosomes, those cells died. So this suggests that exosomes have a sort of detoxification role in our body, which would make sense that they would be involved in these illnesses that they tell us are viral illnesses, because they're really due to toxins, and the exosomes would be part of the healing process. But where I thought this really came into play is, remember I described the experiments before where they mix these foreign cell cultures with antibiotics. Now, antibiotics have been shown experimentally to induce exosomes in several studies. So they basically made a recipe to make exosomes from these foreign cells. So it would be very easy to just snap a picture of that under the microscope and say that it's a virus. And if you didn't actually chemically characterize it and do a genetic sequence analysis, you'd never know what it was. But it would look the same under the microscope. So this was something that I thought was highly probable in the pictures. And when they first started looking at viruses, or what they were looking for viruses actually under the microscope in the 1940s, a relatively short time after the electron microscope was invented, they basically had the same problem, that they would see dying cells that would put out all these kind of particles. And I'm sure some of them were exosomes. They didn't know about exosomes at that time. They've only been discovered about 30 years ago. And then there were other just sorts of extracellular debris and the various types of particles. They could have been apoptotic bodies or all kinds of things. And they realized, the scientists at that time, we can't tell what's what, but we don't find, anything that sticks out as being a unified, thing. And so they were about to give up until Enders came out with that technique of showing the tissue damage that I described before. And that kind of work like a trick. And every single virus paper that I've seen since that time, and that was like, I think 1954 that paper came out, has used this same exact technique to this day. Of course, they added some fancy genetic analysis, which I can talk about, but once again, there's really no process that they use to actually isolate and show a virus with any certainty.
Well, so there you have it. Now, the only thing that keeps going through my head as you're saying it is scamdemic. Absolutely scamdemic. It's the only thing that I can say. And